It is said that an average human can speak up to 4 different languages in their lifetime. This means you can be a translator of 4 types of different code wordings. But the RNA in your body takes the lead. It can create and translate up to 64 different codes. So today, we will discuss how this translation occurs within our cells. We will be focusing mainly on the steps of translation, which are initiation, elongation, and lastly termination. In our last video, we talked about 3 types of RNA. Messenger RNA, which is also known as codon. Then we have transfer RNA, which is anticodon. And lastly, ribosomal RNA is mainly present in ribosomes. Can anyone tell how this RNA was made? They were made by DNA through the process of transcription. If you don't know what is transcription, then pause this video right now and watch our video on cell transcription first. For those who have watched the video, let us proceed. So, we said that there is messenger RNA which will be responsible for providing the genetic codes. Look here, that is how the messenger RNA looks. And the codons are always made in the form of triplets. Hey! Does anyone remember what were the nucleotides made of? Let us remind you. RNA is made of 4 nucleotides: adenine, guanine, cytosine, and uracil. Now let us do some math over here. If there are 4 nucleotides and the codon are supposed to be in the form of triplets, then how many genetic codes will be formed? Well, if 4 has the power of 3, this means there will be 64 codons made by the messenger RNA. So out of these 64 codes, 61 of them are used to start the process of protein synthesis, and the rest of the 3 will stop the process of protein synthesis. Let us dive deeper and make all these 64 codons, shall we? If we make a table of sets, of course, the old math has kicked in. Keep the 4 nucleotides common on 2 ends and makes each of the nucleotide at the last end. Here is UCAG. Then again UCAG. Then on this side write UCAG like this for 4 times. Now start making codon sets. 1 U from here, 1 U from here, and 1 U from here. So the code will be UUU. Then again 1 U from here, 1 U from here, and 1 adenine from here. So the code will be UUA. As you have got the gist, so let me put all the codes down for you. You can create your codes yourself whenever you like now. Why did we go to this much deep level and made a whole chart about it? Well, this is to make you understand two things. First thing about this table is that it has only 3 stop codons, which are UJ, UA, UAG. How will you remember them? Simply by a mnemonic. You go away. You are away. And you are gone. You go away means UGA. You are away meaning UAA. And you are gone means UAG. So, what was the second thing then? The second thing was that only 2 amino acids, methionine and tryptophan, has 1 genetic code. Other than that, all the amino acid arises from 2 or more codes. AUG is the sequence specific for methionine. UGG is the sequence for tryptophan. Remember these 2 things as they are asked in your exams a lot. So, coming back to our triplets in RNA. This messenger RNA has 3 properties that you also need to keep in mind. Number 1 is that these triplets formed are comma-less. This means that this will never happen if the 3 codes are made and then one nucleotide will be left alone and then it jumps to the next nucleotide. They will always form codons in the specific triplet sequence. Number 2 is that these triplets are non-overlapping. This means that this will never happen, that a nucleotide will be taken from the sequence and will be used in another sequence. So, no overlapping. But there are exceptions in these 2 cases when it comes to viruses. So, if someone asks you which organisms don't follow comma-less and no overlapping rule in their RNA, please reply that they are viruses. The third property is that many triplet codes can give rise to a single amino acid except methionine and tryptophan, as we have discussed before. This is done by RNA in order to prevent mutation in the genes. That was about the messenger RNA. Now comes the transfer RNA that will read the codon on the messenger RNA and makes the anticodon sequence for it. This is the typical structure of transfer RNA. This is 5' and this will be the 3 ends. Transfer RNA has 4 loops. T loop, D loop, variable loop, and anticodon loop. T-arm basically tethers the tRNA into ribosomes. Like this. The D-arm or the D-loop is responsible for the identification of transfer RNA by the enzyme known as tRNA synthetase. Variable loop, as the name suggests, that variable proteins bind in here, so it is not important to talk about in translation. The anticodon loop is mainly responsible for reading the codon from messenger RNA and then converting it into its complementary nucleotides. Now the anticodon will also exist in the form of triplet codes that we have discussed in messenger RNA. So, that is how a complex of these 3 RNAs is made. There is messenger RNA. Then this will be transfer RNA. And lastly, ribosomal RNA will be attached to ribosomes. Now this complex will start the process of translation. Translation will have 3 steps as same as the transcription. There will be initiation, then elongation, and lastly termination. Let us go ahead and start with the process of initiation.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.